Great, thanks very much, Chrissy, and thanks very much to the organisers for putting the event on. And of course, big thank you to everybody for taking the time to listen in today. We're delighted to have you here. And so, yes, that's exactly what we're talking about, Arovella Therapeutics. For those of you that don't know us, our ticker is ALA, and we are a biotechnology company focused on the battle against cancer. I will be making forward-looking statements, so please do take note of the disclaimer. Now, I'd like to start with the highlights of the company, and what we're working on we term an invariant natural killer T-cell platform, which is a mouthful, and I'll unpack what that means in a moment, and for short we call it iNKT cells. And we're using that to develop initially treatments for blood cancers, but as Chrissy just mentioned, also looking to tackle a range of solid tumours as we build up from behind. So our lead product is ALA-101. That's going after a very specific form of blood cancer, a leukemia or lymphoma that has something we call CD19 on the outside, and we're expected to take that into clinic in 2024. Now, Steve Utomi-Clark did a lovely job explaining the cell therapy sector earlier, and he highlighted a lot of the key challenges. And so we believe that our iNKT cell platform solves a lot of those challenges, which is why we're so excited by it. Now, we've worked very hard to build an equally impressive team as the technologies by bringing in both board level and management level individuals across Australia and the US. We have built the company through very careful strategic acquisition of technologies from world-leading institutes like the Imperial College London, the MD Anderson Cancer Centre, and we'll continue to do that as we look to grow, bringing in new things that align with our focus areas. We are the only company in Australia working on an invariant natural killer T-cell platform and one of a handful worldwide, so we do see that we're well positioned for growth. In terms of our financials, we're currently valued at around 40 to 45 million. As of the end of June, pro forma cash was just under 7.4 million, with the proceeds from an SPP that was completed early January. That does not include our R&D tax incentive funding, which we expect before November. We've got an excellent register with strong support from our largest shareholder, Merchant Group, but also a number of high net worths and also smaller institutions that have been very supportive of what we're doing. So we've had quite a good start to 2023, good start, I should say, seeing a pretty strong share price appreciation after we announced and presented data at a very well-known conference in the US called the American Association for Cancer Research, but clearly looking to grow the shareholder value as we move forward and take these products into clinic. Now, I won't go through this in great detail because Steve did a wonderful job of describing it earlier. Just to say, this is the typical cell therapy regime where we take the initial starting material from the patient. Then it gets genetically reprogrammed, so it will specifically tackle cancer. It gets grown and given back to the patient. And I'll explain why our technology, we believe, is better than this original, what I'd call almost generation one of the therapy. And again, not to go into great detail, but Steve did a great job explaining that for certain blood cancers, patients are now 10 years cancer-free. So we are using the word cure and cancer in the same sentence. The revenue for each of the first two products is doing quite well, and we're expecting this to be a greater than $60 billion sector before 2030. So as I said, what I'd like to talk about is why our platform actually addresses some of the challenges that have hampered the sector. And just to make it clear, the major challenges are because the therapy needs to come initially from every single patient, every dose needs to be made bespoke. That's very, very difficult from a supply chain perspective, and of course, very expensive. So the dose per patient is about $500,000 US. Also, the starting material is compromised because the patients have seen cancer, also likely chemotherapy or other types of therapies. And not all treatment centres will actually be able to undertake this process, so a lot of eligible patients actually don't get the therapies. In addition, it takes about 4 to 6 weeks for the therapies to actually be manufactured, so people die while they're being manufactured. They don't actually make it to take the therapy. And of course, because you're making it every single time, there's also the risk of a manufacturing run failure. So as I said, we believe that specifically our iNKT cell platform addresses these challenges, but also offers the potential for improved activity. So just to introduce iNKT cells, this is a nice cartoon picture of some of the immune cells that are in our bodies, and we allude to these as the soldiers of our immune system that fight infection or target particular forms of cancer already. But what we're most interested in, obviously, is the iNKT cells, noting that T cells have been most well studied, and that's where all of the 6 approved products derive from. But natural killer cells are another type of therapy that's also been particularly attractive in this sector. So why iNKT cells? Well, they are the front line of the human immune system, so they're very rapidly activated to fight infection and cancers. They actually bridge the two halves, the slow-acting and the fast-acting aspects of our immune system. They contain both T cell and natural killer cell killing mechanisms. And as I said, they naturally target certain types of cancers and actually trigger their destruction already. They've got a very strong safety profile. We can give them from a healthy person to a patient, which is in contrast to the original therapies. And they have multiple anti-cancer properties, unlike some of the other immune cells, where they actually destroy other immune cells that will promote tumour growth. And they also infiltrate the tumours themselves and then send out signals to the other immune cells to come and get involved in the destruction of the tumour cells. Now, we also believe that the iNKT cell space itself is untapped. And so when we saw the sector start off, really, I think it was set on fire in 2018 after two multi-billion-dollar acquisitions, we saw an absolute flurry of T cell companies and natural killer cell companies. And this is a snapshot of those companies in the sector. And contrast this with the iNKT cell space, which is where we play, and there's only a handful of companies worldwide working on this type of technology. Now, in terms of how it actually looks from a manufacturing perspective, so we have two major changes. One is we collect the blood from a healthy donor, a fully functional immune system. We isolate their iNKT cells from that blood. We genetically reprogram them so they have the CAR or the laser-guided missile that will find the cancer cells. They get grown up into the billions before being put into small vials, stored in a freezer, and then when a patient is eligible for that therapy, it'll be pulled out of the freezer and it can be administered. So we believe this has a number of advantages. We have a healthier starting material. It's scalable because we can create multiple doses per batch. People get access to the treatment a lot faster, and of course it eliminates the risk of any manufacturing run failure because it's sitting in the freezer. Now, I would like to just quickly go through some of the data of why we're so excited by this platform. And these are mouse studies that must be completed before taking these types of therapies into clinic. So what we're looking at here is a series of mice. Anywhere you can see the red, green, blue, yellow colour, that's the tumour cells inside of the animals. We can image them. And so what we're looking at here is we've given them a range of different treatments, and we're looking after 3 days. So the first is salt water. The second is a T cell with no laser targeting. iNKT cells also with no targeting. CAR T targeting, CD19 targeting T cells. So these are equivalent of what's approved and on market. And then our therapy in the last column. And so I think it's quite clear after 3 days you can see all of the animals in the control groups, as we call them. They're still quite a lot of cancer cells persisting, but very clearly in our case, the animals have eradicated the bulk of the tumour cells present. So we know they're very quickly activated, but what we're probably more interested in is how long do the animals actually last after they've taken these types of therapies, because that's what we'd like to recapitulate in humans. So this is a survival curve where 100% of the animals alive would have the line up the top. 0% is if the animals have all died, and down the bottom we're looking at out to 90 days. So in all the control groups here we can see the animals have died between 40 to 60 days. The CAR T, so the original type of therapy, we see that it's really good data. 60% of the animals are actually alive at 90 days, which is wonderful. And as I said, this type of data led to their approval by the FDA. But what we see for the iNKT cells is it's about 90 to 95% of the animals are alive. So we see that there's this improved opportunity for activity as well. Now, one other interesting finding that came out of these studies was looking at 4 mice that were actually treated with our therapy. At the earlier time, you can see they all looked like they were free from cancer cells in their body. And between 20 to 30 days, the cancer cells actually came back. And you can see it in all of them, actually. It was localized to the brain. Now, the natural response here is, okay, the animals will die, and our curve that I showed previously won't look as good. Instead, what happened was the animals actually underwent what we termed a spontaneous secondary remission. So they were able to eradicate the cancer a second time. Now, this is quite an exciting finding, and we're exploring this further, but it suggests that the cells can persist long enough to take care of the cancer a second time, also that they cross what we call the blood-brain barrier, and that they have an impact on a more solid tumour-like cancer type. So one thing we're also working on is taking ELA-101 and combining this with Immugene's onCARlytics platform. So they actually have ELA-101, as I've just described, it's a CD19 targeting therapy, and we're planning to use that off the shelf. CF33, or the onCARlytics platform, actually infects tumour cells and labels them with CD19, which our cells will recognise. So we think this could be a wonderful combination therapy. And let me just show you how it might work. We'd inject a human with the oncolytic virus. It would go in and infect the cells before then labelling the cells that aren't killed with CD19 on their surface. And then what we're able to do is actually inject our iNKT cells. They'll come in, recognise the CD19 on the surface of the solid tumour cells, and then trigger their destruction. Now, as I mentioned a moment ago, iNKT cells also influence the tumour microenvironment. They recruit other components of the immune system, so we expect a multi-pronged approach to killing the cancer cells. And for this program, we're expecting in vivo or animal data this half, which is quite exciting. So all in all, we've got a range of shots on goal. We've got ELA-101, we're treating for blood cancers, the OnCARlytics platform plus ELA-101, which opens up shots on goal for a range of solid tumours, and two programs I haven't talked about today, but another targeting CAR or laser-guided missile to target multiple myeloma and other solid tumours. And we've just have an option agreement for another cytokine technology from the University of North Carolina, which will strengthen the whole platform across a range of solid tumours. So we've got quite a busy 12 to 18 months planned. The most important milestone is for us to take ELA-101 into clinical trials next year. And that, as I said, will be against a range of blood cancers that produce the target CD19. We're also continuing to assess novel technologies that we'd like to bring into the business to expand the use of the platform against more cancers like solid tumours. But over the next 6 months, we've got quite a busy level of activity manufacturing the product, which we expect to yield a number of important milestones that we will announce before ultimately manufacturing the product in the first half next year, and then doing the necessary regulatory submissions so that we can take that product into clinic. As I said, the Imugene and ELA-101 data we're expecting to be presented before the end of this year. And as I said, we're looking to analyse additional technologies to add to the platform, and then first half of next year we'll generate additional proof of concept data for those studies as well. So I alluded to it, but we've got an exceptional team with a range of individuals based in Australia and based in the US. And what's important is we've got a significant amount of experience across drug development, right the way through from research and development through to development of drugs themselves, but also commercial activity in terms of the form of a fair bit of corporate transactions, which is great to have. As we look to build the company into clinic, we expect that we're having nice conversations with some pretty important players in the cell therapy sector. So, and just to that point, we are preclinical, and that's fine by us. But what we've noticed over the last 2 to 3 years is in this particular space, the number of transactions that occur in early phase 1 or at the preclinical stages, whether it be an acquisition or license agreements, is still relatively strong, given that the biotechnology sector has had a pretty tough 24 months. So what we can see is this, for example, a more recent transaction in May this year, a phase 1B company, so first trials in humans. The upfront fee for a license agreement was $245 million US. And again, thinking about Arovella's market cap at 45 million Australian, it dwarfs that. And we've seen a series of those types of deals across both phase 1 and preclinical companies. So in summary, we are developing an off-the-shelf iNKT cell therapy platform. We're we're the only one in Australia and one of a handful worldwide. We do that because we know the iNKT cells have multiple properties that will be beneficial. It offers improved manufacturing and logistics abilities, which we think will drive down the price and provide a lot more access to these types of therapies. Our lead product we're expecting to take into clinic in 2024. We do have an expanding pipeline, which we think is very important in terms of strategic partnerships. And for all these reasons, we do think that we're very well positioned for growth and looking forward to continuing to grow our shareholder value. And with that, I'd just like to say thank you all again very much for your attention.